Steve, this is Brian. I just want to let you know I was on the line.
Thanks, Brian. Well, why don't we get started? Thanks for joining us, first of all. This is Steve Fuller. I'm with the Science Applications Program in Region 5. We invited well over 100 people to join us, and most of that group was connected through Robin Niver, and it represents the folks that have contributed to the status assessment process for frosted-elfin. Robin couldn't be with us today. I'm part of a larger team of people that have been working in the background to help Robin stand up the protocol and a survey effort for the region. So why don't we go ahead and get started? I'm going to hand the ball over to Bridget McDonald, who's taking questions and managing the actual webinar. So go ahead, Bridget.
Thanks, Steve. You can actually keep the ball. I just wanted to go through a couple of logistical notes. I'm going to be muting all incoming lines just to prevent any interruptions during the webinar. So just a heads up. You can move to the next slide, please. If you need to unmute your line, press *6. But we're going to try and move questions to the end of the presentation. So if you do have a question that comes up during the call, hit that chat icon in the menu at the bottom. Next, Steve. Then you'll see the chat window, and at the bottom of that, there's a little drop-down menu where you can select who you want to chat. You can send your message to Heather Zacharitz, the meeting host, and we'll monitor that chat window. If applicable, indicate the slide number your question refers to so we can go back to that exact spot in the presentation. And we'll just keep track of all incoming questions and make sure they all get addressed at the end. If you happen to be watching a recording of this webinar and won't be able to ask questions live, you can email your questions to Robin Niver. Next screen, Steve. Okay. So I'm going to mute the call at this point.
The conference is now in silent mode.
So Steve, just hit *6, and that will unmute your line.
Thanks, Bridget. Can you hear me?
Yes.
Okay. All right, so today's webinar is about our effort to launch a pilot frosted-elfin habitat and butterfly survey. Robin distributed protocols, and I also distributed them fairly widely. So today, we don't have enough time to get into the finer details about the methodology, but we do want to help you, understand what's included in the protocols, generally how to use them, and give you a snapshot of what our process will be to record and submit the results. And I just want to remind folks that we'll be doing the same webinar on April 11th, and in follow-up, at the end of the call, we'll talk about some next steps to help folks get up to speed on using the surveys if you want to be included. So I'll be doing some of the presentation, but we'll also be calling on others like Becky Longnecker from our Refuge program, Blake Massey from Ecological Services on the data side, Bridget McDonald from Science Applications has been helping us, and Brian Underwood from U.S. Geological Survey may be joining in to help field questions on the mechanics of some of the survey protocol. And again, this is Steve Fuller with the Science Applications Program. So I'm going to be very brief on the background. This is more or less a one-slide background because, again, we've invited folks that have already been pretty closely engaged in the frosted-elfin status assessment process. The bottom line finding from the 2018 preliminary status report that many of you contributed to is that there are many unknowns and a large-scale conservation opportunity. The ranking process determined that 86% of the historically known populations are currently unknown. Folks haven't been back to survey or monitor those sites in quite some time. I wanted to give a little bit of context, again, since many of the folks on the phone have been involved in the status assessment process. I just wanted to share the larger framework within which Region 5 is thinking about conservation of at-risk species. That includes all species that are under review for listing under the Endangered Species Act. The Science Applications Program, which formerly hosted landscape conservation cooperatives, is now managing at-risk species program, of which species status assessment is a part. So we're working across programs within the Fish and Wildlife Service, and we're doing a lot of work to support landscape conservation partnerships to get pre-listing conservation off the ground. What do we mean by pre-listing conservation? Well, it includes some of the activity of a species status assessment, but it also includes new research and monitoring, like the work we're talking about today. It includes conservation design and planning, which can be a pathway to influence the outcome of a status assessment. It includes on-the-ground delivery of conservation work to hopefully improve the status of species that are under review. So that's the framework we're working with in the Northeast region. And again, you'll hear from a group that's represents multiple programs within the service, and is just the tip of the iceberg for the kind of conservation work we're planning. Okay, so today we're going to focus in on the regional survey effort. We will be reviewing the pilot survey protocols. And again, this is a pilot. We encourage folks throughout the 2019 effort to continue to give us feedback. We've had some great comments and feedback so far. We'll talk about how to collect and submit data to U.S. Fish and Wildlife Service, which is optional, but it's encouraged if you want to continue to be a part of this species status assessment. And we'll also take questions and feedback. We might not get to all of the questions and feedback today, but we will be recording them and we'll do our best to follow up. Okay, so I'm going to hand the ball over to Becky Longnecker. Becky, are you here?
Yeah, hi Steve, thanks. Hi everyone, this is Becky Longenecker. Like Steve mentioned, I'm a wildlife biologist. I work with the National Wildlife Refuge System here in the Northeast region. I'm also our regional pollinator coordinator. And so I just wanted to speak briefly about the overarching survey effort that's currently in place and how the Fish and Wildlife Service is contributing a little bit more manpower to that effort. So to varying degrees, we know that states have been undertaking their own survey efforts for frosted-elfin over the last number of years, and many have plans to do their own surveys in 2019, for example, Texas here. But the service is also dedicating resources to help states fill in informational gaps, particularly here in the Northeast region. Next slide, Steve. So to that end, we're dedicating resources to kind of amplify the survey effort here in the northeastern states. And to do that, we're hiring 10 ACE at-risk conservation fellows this year. ACE stands for the American Conservation Experience, which is a nonprofit organization that'll be doing the hiring for us. And so by hiring those 10 seasonal staff, we will be stationing them in pairs at 5 national wildlife refuges that are spread across the region, and those refuges are shown on the map there in green. They stretch from Blackwater and Maryland to the south up to eastern Mass in New England. And the idea is that each pair of ACE fellows will perform frosted-elfin surveys at high-priority locations in the states that surround those refuges. And we're working in close coordination with state biologists and other partners to make sure that their work is directed to the most significant locations that need to be surveyed, particularly focusing on those historic locations that are currently in an unknown status, like Steve mentioned in the background. And so through this service and state partnership, we're going to be providing additional survey support to at least 8 states, and there may be an opportunity for us to provide even more support to more states. So I would say if you're interested in that and you haven't been in contact with us yet, feel free to reach out to myself or to Steve. And thanks. I'll kick it back to Steve to start talking about the mechanics of the survey protocol.
Thanks, Becky. Okay, so now we'll hone in on what's actually in the protocols. The significance of the protocols, number one, this is an attempt to improve methods for doing surveys for frosted-elfin, not just by looking at populations, but also by doing a rapid assessment of the species habitat. And just a reminder that this is a pilot this year, and so we're really hoping to get some good feedback as folks test this stuff out in the field. Of course, it's significant that we need to reduce uncertainty about the species status. It's very difficult to make a sound decision about listing with 86% of the populations in an unknown state. There's value, we think, in establishing a standardized approach across the region. Of course, we don't expect that everyone can pick up this method this year, but we do hope that we can start to get some traction and get some standards in place range-wide for the species. We've also set up a fairly flexible data submission process, and if folks already have walkthrough counts on the ground, I think going on the ground and have a different some different approaches in place, we can probably find a way to rectify the data and still make a contribution to the status assessment. And last, it's important that we get better on the ground information in order to plan out future conservation for the species. So specific objectives from our end for the survey protocol are, one, to measure frosted-elfin presence and absence, and in the process of doing that, also begin to gather a data set that we can use to estimate the probability of detection for the species, because we want better information about what the absences mean. Second objective is to look at an index of relative abundance. Walkthrough counts are widely used as a measure of butterfly abundance. It's fairly well known that there's an enormous margin of error around those estimates, but nonetheless, it's the most readily attainable measure of population abundance, and it'll be the best that we have available to us in a short time frame. Next, we want to look at habitat extent and quality. Again, a big part of the unknowns for the population status is that we just don't have any idea what the habitat looks like for a lot of the historic records. And last and not least, we hope that there's a useful correlation between the walkthrough count, index of abundance, and what we see in terms of the habitat. So those are our objectives. What is contained in the protocol is essentially a set of 3 standard operating procedures, SOP 1, 2, and 3. The first one is a preliminary site evaluation. This doesn't require field work necessarily, but for locations that are fairly well known. But it does provide a way for us to solidly confirm whether or not a site has potential now or in the future, and it's useful to have that for future planning purposes. The second of the operating procedures is marking out where the habitat patch is and doing the counting of the butterflies. And the third is a rapid habitat assessment. There's also some information in the protocol about timing and some general discussion of the methodology. There's also 3 data sheets included, A, B, and C for each of the standard operating procedures. Those are paper data sheets, but those paper data sheets are also reflected in an Excel workbook with drop-downs and some macros that help with data entry. And there's also a Survey 123 application that we've developed that we'll discuss later in the webinar. So there's several modes of data entry available. So the preliminary site evaluation has value in finding sites that are no longer suitable and for confirming a site's merit for future survey work, whether that's this year or in a subsequent year. And this is important to us, especially if it's been a long time since a site has been visited. Again, this may not require on-the-ground field work, but it could be done remotely if there's some familiarity with the site. It's best to do the preliminary evaluations early in the season when we're just beginning to see the emergence of the host plants, wild indigo or blue lupin. And again, if it may not be necessary to revisit the site if it's been visited in recent years. The preliminary site evaluation information can be entered in paper form on data sheet A. Even if you're not using the paper data sheet, there is some potentially useful explanatory information on it. You know, it is designed to be largely self-explanatory. It does contain fields for entering information about permissions, describing the access, and it contains GPS fields for entering the location of access for a site, and asks questions about prior surveys, all again with the intent to get solid documentation about current and potential sites for surveys all together in one place, and to give clear direction for folks to access a site for future surveys, even if we can't get to it this year. Okay, so just want to do a time check here. It's 1:19. I think we're on schedule. SOP2, marking the host plant patch and conducting the butterfly counts. So the purpose of this SOP is to describe how to conduct the butterfly counts. A key first step is defining where exactly the counts will take place. Another critically important piece is that this is where we will document presence and absence of frosted-elfins, and from which we will derive both the presence, absence, and abundance index. So the timing is critical. Many folks on the phone have been involved with butterfly counts for many years, or have learned enough recently to understand that it's absolutely critical. The flight period for frosted-elfin is anywhere from 3 to 6 weeks. Well, it says 2 to 6 weeks here, but it varies. And if you're not on time, you could miss the peak and really throw off your effort or completely miss the brood. And what we're looking for in this survey is a minimum of 3 counts, spaced roughly 4 to 7 days apart. And the thinking behind those numbers is that it gives us enough spread in time that we increase the likelihood that you can capture the peak number of butterflies. And by repeating the surveys, we also, again, begin to build that data set from which we can estimate the likelihood of detection. We're looking at initiating as early as mid-February in the southern states to late May in the most northern states. And again, there's a lot of variation around that. and we recognize that many states, especially further south, are probably underway or even beginning to wind down their survey season for frosted elfins. And just a footnote there, we had intended to be having this conversation in very early February, but due to the government shutdown, we were delayed by a couple of months. Okay, so a key part of the protocol for conducting the butterfly counts is to establish the count route. So we're looking to establish a fixed beginning and end point for the surveys each year that the survey is conducted, and that's because the host plant populations change from year to year. And what we want is a beginning and end point that allows the observers to repeat the surveys throughout the season and consistently capture the population of host plant. To get a good fix on where to put the beginning and end points, it's important to allow the plants to begin to emerge and to mark semi-permanently, ideally with a wire stake flag or something of the sort, at the beginning and end point of the host plant patch. The paths will vary throughout the season. That's okay, but it's important that each path attempt to cover the most dense parts of the population and begin and end at the same location. So when an observer begins the survey, they'll obviously begin at the marked starting point and attempt to visualize a counting path that covers the whole patch of habitat. They'll walk slowly but at a steady pace, visually observing the butterflies. They'll try to weave through the patch and pass by the densest, most densely populated areas populated by host plants, that is, and they'll tally the butterflies without stopping. Surveys should be conducted under optimal conditions, ideally 65 to 95 degrees Fahrenheit. They should be conducted when it's not raining and the vegetation is not wet. Wind speeds should be less than 20 miles per hour. And if the temperature is less than 70 degrees, ideally it should be sunny and the wind should be even lighter. Under cooler temperatures, if the other conditions are optimal, then you may have some success. So for the SOP2, the data can be entered on data sheet B. On the top half, we have some fairly standard observer and event information. I want to point out that on the right side of the top block, there's state, town, site name, patch code, and ELU code. Ideally, we can get all of those identifiers in there, but we recognize that everybody may not be using the same kinds of identifiers. So make sure you look at the description in the protocol and enter the appropriate ones. In the next block, we have environmental data. There's a lot of different ways we can collect environmental information. Ideally, we'll use handheld thermometers to get the local temperature. If that's not available, we've put a spot in to enter a more regional site temperature from a weather station or something similar. The other fields, we've tried to make simple categories and eliminate as much as possible measurement. These variables are really here to just ensure that we're within appropriate windows for surveys. And also, the environmental conditions contain some information about threat and conservation security. again, these are just indicators to help us understand the status of the species. The bottom half contains the count data and some fields to clarify how you're verifying the identity of the species. So we'll first enter the start time, go ahead and do the walkthrough, count the butterflies, and then enter the end time. The time spent is a potentially important covariate for estimating detection probability, so it's pretty important to gather that information. We also have, can't get rid of this box, the information about how adults were identified and more information about the other life stages that have been observed. And it's not a hard requirement to document other life stages, but as most of you are probably aware, it can be difficult to identify butterflies on the wing. And with practice, some of the other stages can provide good supporting information. We don't need to get photographs in hand, but it's certainly useful for you to take photographs and keep them available in case there's any question about the identity. And again, in some states, there will be some verification requirements that the state has, and we want to make sure the information are available to satisfy those. And last on this sheet is looking at the patch location information so that we can relocate the patch. Okay. My next button isn't working very well. There we go. Okay, so moving on to SOP3, which is the rapid habitat assessment. The purpose of this, obviously, is to rapidly assess the quality and abundance of the habitat. We want to know how important is the site within the overall range-wide context of the species, and is there a management need or opportunity available. This information can also be used or will be used to take a look at potential correlations between the habitat and population size. Sorry, I'm going in the wrong direction. There. So within SOP3, the first section looks at delineating the habitat patch. If you'll recall from SOP2, we are recommending to make permanently, well, semi-permanently mark the beginning and end point of the walkthrough count, and that will generally mark at least one dimension of the habitat patch. But we're recommending that when the host plants have fully emerged, ideally in the flowering stage, that we'll actually use one of several techniques to draw an imaginary line around the patch and estimate the vegetative cover area. We're also looking at the cover of different classes of vegetation, documenting which host plant species are present. In some, but not all locations, there could be 2 or more species of host plant, potential host plant, in a single location. We want to know what's there. And then we're looking at host plant abundance information, which is really a key covariate with the population size. So defining the patch is one of the key steps in this process. How we do it is important. We want to be as consistent as possible, but this doesn't need to be perfect. We just need to be somewhat consistent. So what we want to do is draw a convex line around most of the plants. We want a roughly consistent coverage of the host plant within the delineated area, but again, it doesn't have to be perfect. It's going to be, some unevenly distributed plants, and that's fine. We don't want to go out of our way to try and capture every single plant. So on the left, there's a convex line that's been drawn around a patch and excluded a few outliers. On the right, one line attempted to capture every single plant, and it leaves a lot of empty space, which potentially skews how we view the abundance of the plants. On the right, it's a very jagged, not convex shape, and that's also not desirable for our estimation purposes. Again, the whole purpose of drawing a line around the patch is to estimate an area. And the area, coupled with some of the other measures of abundance, plant abundance that we'll talk about, gives us some real grounding for estimating the plant population size. Okay, so there are several methods of measuring the habitat area once you've visually drawn a line around the patch. We're recommending on-site. You can do a pretty good job of this on a smartphone right in a GPS application like Google Earth. I've tested it several times using Google Earth, and it works quite well. The protocol document has fairly detailed instructions on each of these methods, and so have a look there. Again, the purpose of drawing a line around the patch is to estimate the area. And that, after all of the effort to visualize a line and then find a way to document and estimate the area, it just results in one habitat area number being entered on the data sheet. The habitat assessment section also contains a number of visual estimates for canopy cover. There's some instructions on there, some definitions for each of the classes that should help. We're just looking for a coarse estimation of each vegetative stratum that could influence the condition of the habitat, especially canopy cover. When the canopy starts to close over, we may lose the host plant, and so we want to get an estimation of that. Measuring the host plant species that are present and which one is dominant and the likely actual host plant in use. And this is done just by circling on the data sheet. And to make estimation of the host plant abundance simple, we're testing out a couple of methods that are somewhat unorthodox. We've consulted with several people that have done a lot of survey work for wild lupin and blue indigo, and tried to distill an approach that will make it simple. Again, we're looking for a coarse index coupled with an estimate of the area. So we want to keep it simple. It's notoriously difficult to measure plant populations quickly, and so here's what we've come up with. The first approach is a visual density estimate. Again, looking in the protocol document, there are actually 5 classes, A through E, A being the most thick and E being the most sparse, and there are several different kinds of patterning shown in each of those levels, and we're just looking to pick one of the rows or one of the classes, A, B, C, D, or E. That's all that needs to be entered on the data sheet. The second estimate is a walking density estimate. Again, this is intended to be a pretty quick and dirty way to get a handle on the density of the plants within the mapped area. intuitive and tied to how folks actually experience the plant population on the ground. So this is, essentially how easy is it for you to walk through this patch of host plants without stepping on the plants? Is it impossible to avoid stepping on them, all the way down to E, where you can pretty much waltz straight through the patch and stay a couple or several yards away from the plants at all times, or anywhere in between. Look at the protocol. The descriptions are also included on the data sheet so that you can make a comparison and pick the one that best represents your experience. And last but not least, using handheld counters to actually count the plants. We set up a fairly simple exclusion rule, or two exclusion rules. If a plant would fit inside of a milk jug, is slightly larger than what would fit inside of a milk jug, or if it has a flowering stem, then it should be included. Everything else, the plants that could be completely covered by a milk jug without touching any vegetative part, and that have no flowering stem, should all be excluded. Again, from folks that have spent a lot of time measuring these specific plant species, when there's a continuous cover, it can be very difficult to separate one plant from another, and so really there's no choice but to revert to counting some other unit, either square meters or perhaps a 3x3 meter, and using a multiplier to scale it up. So we estimated that with the milk jug rule, and assuming some plants could be larger than milk jugs, 20 plants per meter square, or 200 per 3x3 meter plot was about right. And again, these translate to categories, so a precise number is not needed. I just wanted to note on the data sheet, when we were creating this presentation, we noticed on data sheet C that there was a typo in the area shown by the red box. It says described above. Well, things got moved around, and that's actually described below. Okay, so we are just about at 140, which was right on target. So let me just do a quick recap here. SOP1 is a one-time evaluation, and it should help determine whether or not surveys should proceed under Protocol 2. SOP2 is a repeated butterfly count using a path with a fixed starting and end point, but with potentially variable walking path in between those points. We're looking for a minimum of 3 visits. Now, that won't be possible on all sites. A single presence and absence counts is better than nothing, but to really be sure that we capture the peak and that we get a good data set in hand for detection probability, we'd really like to see 3. And then last, SOP3, again, during the season, it's a one-time assessment, ideally during the peak of the flowering for the host plant, and it's a bit later in the season than you would typically begin the butterfly counts, as the butterflies typically emerge right as soon as the host plants emerge from the ground. So thanks for hearing about the surveys, the protocol. Hopefully this will give us information about each site, but also give us a better picture of the overall species viability. And now I'm going to hand it over to Blake. Blake, are you here?
Yes. So I will jump in here for my slide. and talk about the data collection and submission process. That should show up here. Okay. So we know people have different preferences on how they collect and submit data. So we developed two options for both data collection and data submission. The first one here for data collection is just the printed PDF forms, good old paper and pen, take it out into the field, fill it out there. For data collection, the other option that we're giving people is using Survey 123 for ArcGIS, which is basically a digital form that you would have on a device like a phone or a tablet, or actually you can use it now on your laptop. So that's a digital form of filling out one of these surveys. For our data submission, we're also going to use two different options, Excel worksheets. Everybody who's ever dealt with data has dealt with Excel, so we feel people are comfortable using that. And so that's going to be one of the options for data submission. And then the other one is the other component of Survey 123 for ArcGIS, which is actually being able to submit that data virtually instantly once you've finished filling out the digital form. So just to jump in, the PDFs, you already have seen some of this. Steve showed you what these look like, so take these out into the field. Obviously, the one thing he mentioned that's worth keeping in mind is when you go out there, it's good to bring a GPS or some other device to be able to capture GPS locations, because you won't have that necessarily. Once you fill out one of those forms, you will then transfer that data to these survey Excel workbooks that we've developed, and it's basically a one-to-one, so every field on the data sheet corresponds to one of the fields, the columns in this case in the Excel workbook, and every row is a record. And you can also see down there at the bottom there are three tabs: preliminary site assessment, frosted-elfin patch count, and the habitat rapid assessment corresponding to the data sheets A, B, and C. And so this is, again, very straightforward for anybody who's dealt with data entry before. We've got some drop-down menus in there and some data validation checks, and as Steve mentioned, some of those require some macros running in the background. So it is a macro-enabled workbook. If you have any warning that pops up about enabling macros, go ahead and do that. That's going to help you enter that data and make sure it's correct before you send it. And then after you've finished it, you're going to send it to Renee here at the regional office. So there is her email, but it's also in the protocol, so check with that. Next, I'm going to talk about Survey 123 for ArcGIS, which some of you may have heard about at some point, but basically it's a simple form-centric data collection GIS app. It requires an organizational account to allow you to log into the app and download any survey forms that have been shared with you. And I'm just going to go to this website and show you what that looks like. So this is where you can get it. It's available for basically any device at this point. It is available for, iPhones, for any Android devices, and like I mentioned, on your laptop. So this is what you would be downloading as the Survey 123 Field app. And I will show you what that looks like. So this is just my desktop, and this is what you would see when you first load it up after you log in. Again, I mentioned that organizational account. If you need one, contact us. We can set up a collaborator Fish and Wildlife Service account to help for this project, and that will allow you to see all the surveys. And so once you're signed in through that organizational account, and again, contact us, we'll help you through that process, you just click on Get Surveys here. I have a lot shared with me, but just to show you that if you have a similar situation, just to get the ones that you want, you can use this little search bar at the top. And so I will very quickly here download the three surveys. So this is, I'm now set for the season as far as having my surveys. And now this is what I would have on my phone or on my tablet. It would look exactly the same. The dimensions might be a little different, but it's exactly the same interface. And so once you click on this frosted-elfin site assessment, you can go ahead and start collecting data here. The data is organized into different groups of the sections here, so you can see how those are all split out. So just to very quickly kind of show you how some of this works, you know, I can start filling in my information here. And you can obviously do this on your phone. Everybody is now familiar with that. But there's also fields that there are different types of data that we've helped you by doing things like pre-populating drop-down lists so that you can just select the information that you want, but in case, you know, just to show you, I could also do other, and then this would automatically identify other affiliation. So there's a lot of real smart stuff going on behind the scenes here that will assist you in filling out information. This is all, you know, collapsible, and then you can continue to open and close those as you fill things out. One of the really great things here is using this map-assisted location entry. So if you're out in the field and you've got this on your phone, you can go here and click on this. And I'm sitting in an office in Hadley, Massachusetts, so I don't have a GPS hooked up to my laptop. But if you did, this would take you to wherever you are. I have it set up so it goes to Washington, D.C., and I'm just going to move this down and point this at the southwest corner of the Washington Monument. And this is giving me the lat long down here, and all I have to do is click this little check mark, and now that position is stored, and so that position is associated with this data record. So I could continue filling out this survey. So yes, we visited it there before. There's some multi-select fields. You know, this is definitely probably going to not be the best site. You know, paved development definitely is going to be an issue. You know, great views. And I can even do things like add a picture, so everybody can see what I look like here. And so now, as I complete this data sheet or this survey, all I have to do is click on this little check mark at the bottom right to finish up. And now it'll ask me if I want to send it later, which maybe I would potentially do that if I was out of cell phone coverage, continue the survey, or send now. I'm going to go ahead and send it now. So this data is being sent. So I've collected the data, and I've already sent that data. This down here tells me that I've already sent data out to the regional or organizational database here. And I actually have a map that will show that new data. So this just popped up because this is what I just filled in. And just to show you, here's that picture that I just took. But all that other information that I filled in is also all in here already. So the data has been collected and submitted very quickly here in, you know, just a matter of a couple of minutes in this case. Obviously, it's not perfect data or realistic data, but it is showing you how fast it is to actually do this process. So going back to finish this to full screen. I have sort of shown you this little box area down at the bottom here. So filling out that PDF form, converting that data over to an Excel sheet, and then sending that to Renee. That's going to get into our regional database here and our data management process here. The same thing is possible using Survey 123 out in the field to get that data into the regional database management system here. So with that, I'll hand it over to Steve again to finish out.
Thank you. Could someone give the ball back to me so I can continue from my screen? Okay, so I just wanted to give a few more comments. Can everybody hear me? Oh, yeah. That's right, I'm on mute. Thank you. And Bridget, can you see the data management summary screen?
No, are you sharing your screen? It's that the arrow at the bottom, the arrow icon.
Yes, thank you.
Yep.
Okay, are we on the data management summary screen now?
Yes.
Thank you. Okay, so thanks a lot, Blake. There's a ton of great work that's been done to try to make the data management as simple as possible and to give several different approaches for submission of data from the field. But I just wanted to point out a couple of other steps in the process that may happen behind the scenes or not so behind the scenes for some. Once the data have been aggregated from the field, before the service can feel comfortable including it in an update of the species status, our ideal is that states can provide some verification that the points are legitimate. It's actually a requirement for us to coordinate closely with states on species status assessment, and you have better knowledge about the validity of the sites and the data that are coming in than we do very far remote. So once the data have been aggregated in the field, we'll provide an opportunity back to state folks to verify that what's been found in their states is legitimate. And once we have that verification, we'll repeat several more steps internally of quality control and take the measures necessary to get the data cleaned up. And with that, it'll feed into a final regional database for this year, and we'll be engaging Robin Niver, to implement the status assessment ranking system that the status assessment group came up with to rank the species status. So from the regional database, those ranks will get populated and contribute back to a future status. Okay, so now is the time for the summary and some questions. Hopefully the questions have been submitted to Bridget throughout the call. We'll do what we can to answer them. We have a few minutes left. Before we hit those, I want to touch on a couple of next steps. One critical one, Blake mentioned it several times, but I want to make sure that we get this out there. You know, folks are welcome to use this protocol. We hope you do, but in order to submit the data to us, it requires some planning and preparation on our end, and we have a few checks that we want to put in place. We want to make sure that data that we're receiving is being provided in conjunction with the state, if at all possible. But we also want to be able to identify all of the contacts that are providing data in advance, so that we can update you if there are any changes to the submission systems before you get on the ground. And we also, if need to make sure that there's all of the access permissions are in place for accessing Survey 123 and later on to check the data that are entered. So please email Renee Farnsworth at U.S. Fish and Wildlife Service. The email address is on the screen. Her address is also in our protocol document. So this is a critical step for us to get started with the submission. Again, anybody can use the protocol, but to submit it to us and ensure we receive the data, please take this step. So a few more next step comments. We're holding another webinar, an overview webinar like this one, April 11th. And subsequently, we're anticipating providing a weekly webinar at the same time to focus in on certain parts of the protocols. Some of that follow-up will be based on the comments we received from you today. So if you want to hear more about a certain part or get into some discussion about part of it, definitely let us know. Get that in the chat window, and we'll be back each Thursday, at least some part of our team here to continue to provide support. Also, coming soon, outside of this Thursday 1 o'clock time, we're going to provide a much more detailed data collection and submission training. There's some neat technological tools there, but with technology always comes a need for some training and support. So we'll come back and get into that in more detail. Blake was barely able to, scratch the surface today. Then later on in the season, when we circle back to the states and ask for a quick check on the data, we'll reach out and work with you on that process. Okay, so Bridget, I don't know if we can turn off the mute or how you want to handle questions.
Hi, Steve. I have one question so far in the chat box, so I'll ask that, and then maybe we can open the phone lines. So this question is from Betsy from Pennsylvania Natural Heritage Program. She says, is it okay to invite a few citizen scientists to the next webinar who we expect will be helping us with the surveys? If so, how should we handle the invitation?
The answer is yes. You can go ahead and forward the invitation. Our only limitation on these webinars is the size of the account. And I'm not sure, Bridget, do you know the hard number on that?
I think it's 100 for the line. This webinar was recorded, and we will record the next one too, just in case anyone can't access it. And right now I'm putting in the chat box to everyone that link to our new frosted-elfin website. There's a whole page on that site dedicated to the survey effort where you can find the protocol, and it also has links to the webinars. After the webinars are, after we get like a transcript for that, we'll upload the recordings to that page as well. There are a couple more questions now. So from Casey Holsworth asks, is there any indication of a break point between deciding to include a fairly large area as having suitable, parenthetical, in my case, lupin, but not overly dense vegetation versus breaking that area up into multiple more dense patches?
We don't have a hard decision rule for making that determination. And we don't really have a need for one. Trying to delineate an area of somewhat consistent density is all we're after. I think there is some utility in not trying to tackle a vast area all at once with one count. I think we'll get much better results by breaking it up. If you wanted a rule of thumb or more discussion, I mean that'd be something we could handle on another call and just hear what other folks have to say about it. But I think that a distance of if you get beyond 100 or 200 meters from the beginning to the end of a walk-through count area, I think that's starting to be pretty large. So if you're covering a greater airport than that, such as in a more of an open prairie type of setting or on an airport, I think it would make sense to begin to break it up.
Okay, another question that refers to slide 23, I think, or it says, in SOP for description of ground cover, page 23, your options are bare soil or vegetation/litter. Can there be another category? For example, we have host plants growing out on gravel roads. My feeling this may be a sink. Can pupae even reach the soil? It would be interesting to categorize this. Sorry if I butchered that question.
Bridget, could you just repeat again? sorry, I know it's a longish question, but I was trying to get back to the slide.
Or if you want to unmute yourself and ask the question more articulately than I read it, just press *6.
We don't hear anything yet. And
oh, sorry. Can you hear me now?
Yeah.
Yes.
Super. Okay, so my question, so hopefully I can be more clear. I have patches of baptisia that are growing out of roads, so it's a foot of gravel. So how on earth would I categorize that in this SOP?
And I mean, according to this, the area that's occupied by the plant, even though there's little detritus or really proper soil, the area is occupied by herbaceous cover, so it would be in the grass herb cover category.
What about the underneath? Sorry, let me pull up the SOP here. The actual PDF of the SOP, which is on page 23.
Okay, so that would be a case where they're not mutually exclusive.
Yeah, I'm looking at the ground cover categorization. 0 to 10, host plants have negligible bare soil around them. 11 to 30%, they have mostly vegetation with some bare soil nearby.
Yeah, so they're not mutually exclusive. So you could estimate the area of the herbaceous cover, which would include the host plant, and then since the area beneath has little detritus and no other vegetative cover, you could include it in the bare ground class.
Okay, and the one kind of what I'm kind of getting at here is I feel that these patches of host plant that are growing through gravel potentially might be a sink. You know, any pupae that's trying to find its way to bare soil, I can't imagine it's going to find that. So I guess I'm looking at.
Yeah.
I wouldn't consider that quality.
How big? I mean, that seems like a potential threat.
Yes, it is.
That the plant is growing in a substrate that could prevent the larvae from easily pupating. And that said, larvae can cover quite a distance to get to a substrate that they can shelter in. So I don't think we have that particular threat covered. Let's see. Yeah, I think you could just, I mean, that would be an example of a unique one that you could list and describe under the threats, but it's certainly something that's worth noting, and, you know, potentially indicates a prescription for restoration to get the host plant established out beyond the gravel shoulder.
Cool. I will be sure to note that in the threats section then. Thank you.
Thank you. Good question.
It looks like Kathy O'Brien wants to ask a question. Feel free to press *6.
Can you hear me?
Yes.
Yes. Hi, Kathy.
Hi. My question was related not to what you just presented, but what was in the stuff that you sent out. You said that photographs would be looked at by, photographs of larvae and eggs would be looked at by experts. And I was wondering who those might be. And I was wondering, I know that one of the things that you sent out had it telling the difference between frosted-elfins and another species on lupins. But what I'd really like to see is something that tells the difference between frosted-elfin and Karner blue larvae. We see them a lot on pods, and I see them all over the place, and I really couldn't tell the difference between them myself. And I wonder if you've got that resource somewhere as you can send to everybody.
At this point, we don't have, I don't have something that we can confidently point to discriminate between those two larval species. I think there are people out there that could consistently do that, but as far as training up, any workforce to reliably do it, at this point, I don't feel like we have the tools to do it. And if there's somebody available that has that, that would be great. And so that's why we're suggesting taking photographs. As far as getting experts to verify identity, we'll be providing as much training as we can in the Northeast region, but getting species identification instruction is a little bit beyond the scope of this protocol right now. If it ends up being a real barrier range-wide, then we would certainly provide what support we can to get together a training and get identification expertise out to everybody. But we wanted to have the records available so that when we go to states for verification, there's information there, and to the extent that we can, we do have some in-house expertise on butterfly ID and looking at the weight of the evidence, an adult photo, and anything we can get on eggs and larvae. When possible, we feel confident that we can get a decent ID. Some states will actually require a photo voucher.
See, there's one more question in the chat from Casey Holsworth. Does Survey 123 only have to be connected during download and upload times, or constantly?
Only during download and upload. Once you have the form on your device, you can take it out of the field and you could be anywhere where there's not cell phone coverage and enter all the data.
Okay. If anyone on the phone line has a question, you're welcome to press *6 and unmute yourself, or again, enter it into the chat.
And one last, parting comment. If there are, needs for more training or technical assistance, please describe them to us. I think the best point of collection for that kind of request is Robin Niver. We provided her email earlier, and I think. Well, since she invited most of you, I think pretty sure you know how to find Robin. It's not out of the question to organize specialized, focused webinars on particular topics, or to arrange an on-site training if there's enough of a demand and enough time to plan that. And if there's no more questions, then thanks everybody for attending and participating, and we look forward to the beginning of a collaborative effort to conserve this species over the next few years. Thank you. And goodbye.